Hey everyone, welcome to Exam 3 review for biology for non-science majors. We're talking about the biology concepts and investigations, Hoefnagels book, chapters 7 and 8. So let's go ahead and start with this exam review.
Dr. D, 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 Dr. D. Explain stuff.
All right, welcome back. This is Exam 3 review. Exam 3 is only going to cover two chapters, chapters 7 and 8. So let's go ahead and start with chapter 7. This chapter covers DNA structure and gene function. All right, as always, please remember, try to study all the handouts and the lecture that we covered in the videos. I'm going to try to touch on most of the important topics in this exam review, but I might miss something here or there. The best thing to do is obviously, study all the material, but I'm going to do my best to prepare you in this video. So again, don't forget that DNA and RNA are nucleic acids made up of building blocks called nucleotides. You should definitely know the three parts of a nucleotide. A nucleotide consists of, remember, a phosphate group, a pentose sugar, and a nitrogenous base. Very important to remember that. And then do you remember another very important thing is being able to compare DNA and RNA? Remember that the pentose sugar of DNA is called deoxyribose, and the pentose sugar of RNA is called ribose. You remember that the nucleotides of DNA are A's, G's, C's, and T's, whereas the nucleotides of RNA are A's, G's, C's, and U's. There are U's in place of T's for RNA. You should also know that when these nucleotides are strung together, they form double-stranded or double helix structure for DNA, whereas it's a single-stranded structure for RNA. Remember, I might ask you to compare and contrast DNA and RNA on the exam. Another thing to remember was complementary base pairing. Do you guys remember the complementary base pairing? You should know that C's complement and pair up with G's. And you should know that on DNA, T's complement and pair up with A's. Whereas on RNA, do you guys remember what do A's pair up with on RNA? Can you beat Wicket? That's right, as always, Wicket. Wicket says on RNA, A's pair up with U's. Okay, do you remember that? Very important stuff. And do you guys remember when I told you that DNA is directional? DNA, every strand of DNA has a 5' end with what hanging off? Can you beat Wicket? What group is hanging off at the 5' end of a DNA strand? That's right, Wicket, a phosphate group. Very good. You should know that every strand of DNA has a 5' end with a phosphate group hanging off, and at the other end you have the 3' end with a hydroxyl group hanging off. Okay, and let's see if you can beat Wicket on this one. Do both the strands of DNA, do they go in the same direction? So are they parallel? Or do the two strands of DNA go in opposite directions? Are they anti-parallel? Can you beat Wicket? That's right, Wicket, as always. They are anti-parallel. Very good. The two strands of DNA are anti-parallel. All of these things are good to know for the exam. Okay, and remember complementary base pairing between C's and G's, A's and T's. Okay, remember that DNA forms a double helix. DNA forms what's called a double helix. Here's the G's pairing up with C's, T's pair up with A's. Very good. You should definitely know the main purpose of a gene. Remember, genes are DNA sequences that encode specific proteins. So the main role of a gene is to code for a specific protein. and you have chromosomes. Remember, chromosomes are made up of DNA and histone proteins, and together that's called chromatin. Remember, chromatin makes up your chromosomes. And do you remember gene expression has two parts? You should know how gene expression works. The first part is called transcription, and you should know during transcription, that's where the mRNA is synthesized. Then during translation, the mRNA is used by ribosomes to make proteins. And do you remember clicker questions might actually appear on the exam? So let's answer this one together because it might appear on the exam. Clicker question number 1, what is the main function of DNA? We just got done talking about how it's encoding proteins, so that should be your answer right there. Awesome. So let's move on to transcription. Do you guys remember transcription is where a gene is copied to RNA? And here's one thing you should know. Let's see if you can beat Wicket to this answer. Are both of the DNA strands read and copied into mRNA on a gene, or is only the template strand copied to mRNA? That's right, Wicket. Only the template strand. You should know that. You should know that. Like, look at this. See? Here's double strand DNA in blue, right? Here's double strand DNA in blue. This is transcription. And remember this enzyme called RNA polymerase. It's going to read the gene. But look, notice this. Look at this. It's only reading the template strand on the bottom. See? It's only reading the strand on the bottom. It's not reading the non-template strand. So you should know that for the exam. You should know that during transcription, only the template strand, not both strands, only the template strand is read on the gene to produce mRNA. And in which direction is mRNA produced? You should know. mRNA is made or synthesized 5' to 3'. You see this right here? That's what it means. mRNA is being made in a particular direction. mRNA is synthesized from 5' to 3'. You should remember that for the exam as well. Now you should know that in eukaryotes, the RNA needs to then be processed. Once the RNA is made, once that mRNA is transcribed, we need to process the mRNA. And you should know for the exam, you should know what are the 3 mechanisms that occur during RNA processing. Can we remember them? Remember, you should know during RNA processing, there's addition of a 5' cap to protect the mRNA. There is addition of a 3' poly A tail. So see, the 5' cap means a cap is placed at the 5' end of the mRNA. And on the 3' end of the mRNA, a poly A tail is placed, and that protects the mRNA as well. And do you remember what was the third modification? What was the third processing event on mRNA? Do you remember? Can you beat Wicket? It has something to do with the exons and introns. Remember, introns are non-coding regions of a gene. So what do we need to do with the introns before we can do translation? Can you beat Wicket? That's right, Wicket. The introns need to be excised. Remember, the introns need to be removed, and that's why the mature mRNA is shorter than the pre-mRNA. Right here you can see the introns are sequences in genes that are not used for making of proteins, so the introns are removed. That means the introns are excised. That's what excised means, removed from the mRNA. And now the mRNA is ready for translation. Remember, translation requires ribosomes. And again, this is clicker question number 2, and this question may appear on the exam. If the DNA template strand has the following sequence, what would be the nucleotide sequence of the complementary RNA molecule produced in transcription? So if this is on the template strand of DNA, that means when you're making mRNA, remember A's pair up with what? A's pair up with U's. So we're going to complement an A with a U. And the only answer choice that has U is E. So it has to be E. The answer is E. A's get copied into mRNA as a U. The G gets copied into a C. The T gets copied into an A. The C gets copied into a G. And the two T's are copied into two A's. So the answer is E. Remember, mRNA uses U's instead of T's. That's why you have this U here, where the little starts with that. And here's a mastering concept. How is mRNA modified before it leaves the nucleus? Remember, 3 ways, right? The 5' cap, the 3' poly A tail, and excision of the introns. You should remember that on the exam. And now you're ready for translation. Remember that during translation, codons, which are 3-letter words, codons are translated into various amino acids. And do you guys remember the codon table? Obviously, you don't need to memorize this entire table, but I do want you to know I do want you to know 4 things. I want you to know the start codon. Do you guys remember the start codon? You should know on the exam that the start codon is AUG. And you should know what the start codon is. The start codon is the codon, AUG, where translation begins. And it's found where? Is it found at the 5' end of the mRNA, or is it found closer to the 3' end of the mRNA? Can you beat Wicket? That's right. He's always right. It's found at the 5' end of the mRNA, right? So the AUG is found near the 5' end of the mRNA. That means translation begins near the 5' end of the mRNA at the AUG start codon, and you should know which amino acid the start codon codes for. Remember, look at this, it's methionine, or M, okay? You should know that the start codon also codes for an amino acid, methionine. And you don't need to memorize the different stop codons, but you should definitely know that there are 3 stop codons, 1, 2, 3. And any one of these 3 can stop translation. Okay, so you should know there are 3 stop codons to choose from. And do you remember that tRNA are the equivalent of printer cartridges? The analogy I use is that tRNA serve as printer cartridges for translation. The ribosome is used during translation. It's the printer, right? It prints proteins. Remember that the start codon, again, the start codon is near the 5' cap, and that's where translation initiates. And every protein should start with the amino acid methionine. And this continues until you reach a stop codon. I'm probably not going to include this clicker question because it requires a picture, so let's skip that one. And do you remember the purpose of gene expression? Why is gene expression regulated? So this means when a gene is used to make a protein, why would you turn that gene off? Why would you do that? Remember, it's to save energy and to save resources. And lastly, do you recall mutations? You should know that a mutation is a change in a cell's DNA sequence. So a gene, if you change the DNA sequence of a gene, that's a mutation. And do you remember we talked about a bunch of different point mutations? What I want you to know is this. Do you remember when we talked about sickle cell disease in chapter 7 when we talked about sickle cell disease? Now, look what happened. Do you remember that GAG became mutated to GUG, or should I say CTC became CAC, right? You should know what type of mutation that was for the exam, okay? So let's see if you can beat Wicket. If you get it right, what type of mutation? And point mutation is true, but what type of point mutation was this? Do you recall? It was a missense mutation. That's right. And do you recall what a missense mutation is? A missense mutation is a type of point mutation, but it's one where you change a nucleotide. And isn't that what we did here? We changed a nucleotide CTC to CAC. You see that? So we changed one nucleotide. So you should know, again, that sickle cell disease is caused by a point mutation, but more specifically, it's caused by a missense point mutation. You should know that for the exam. Okay, and that leads to the end of chapter 7. I tried to remember those most important points for you to remember on the exam. Let's take a quick break time and see what Gizmo and Wicket are up to. We'll be right back for chapter 8 to finish off this exam review. All right, welcome back from break time with Gizmo, Wicket, and Dr. D this time. So, Chapter 8, DNA replication, binary fission, and mitosis. Let's go ahead and get started. Remember that all cells divide, and remember that mitosis is how eukaryotes, multicellular eukaryotes, it's how we grow, develop, repair. But meiosis is the process by which we make our gametes. Remember what the gametes were? The sperm and the egg. So remember that meiosis results in the sperm and the egg. Mitosis results in growth, development, and repair. Remember the term apoptosis? You should know that apoptosis refers to programmed cell death. And here we are at clicker question number 1. Shortly after fertilization, a zygote, remember a zygote is a fertilized egg, divides into 2 identical cells. What type of cell division produces these 2 cells? Remember that was mitotic cell division. Remember, meiotic cell division results in the gametes, the sperm, and the egg. Apoptosis results in cell death, so the answer has to be A. Remember that the genome includes all of an organism's chromosomes, all of its genetic material. And then we want to remember the process of DNA replication. Remember how DNA is copied. You should know the jobs of each of these different DNA replication proteins. So you should know, for instance, you should know all of this. You should know that helicase unwinds or unzips the DNA. You should know that these things are called single-strand binding proteins. Single-strand binding proteins stabilize each strand. Remember what they do? They prevent the DNA from reannealing. You should know that the single-strand binding proteins prevent DNA from reannealing. You should know that primase or RNA polymerase adds a short RNA primer. It's what puts down the RNA primer to the template strand of DNA to start copying that strand. You should know that DNA polymerase adds nucleotides to form new strands. And lastly, you should remember that ligase forms covalent bonds between adjacent DNA segments, or Okazaki fragments. Remember, you should remember the term Okazaki fragments. Ligase binds Okazaki fragments together on the lagging strand. Again, you should know that the leading strand during DNA replication, the leading strand is building DNA towards the fork, right, in a 5' to 3' direction towards the fork, while on the lagging strand, the DNA is being built in a 5' to 3' direction away from the fork. And that's what results in all of these Okazaki fragments. On the lagging strand, you have Okazaki fragments. And remember, these Okazaki fragments are made up of primer and DNA, RNA primer and DNA. Remember that eukaryotic chromosomes have multiple origin of replication. And do you remember how DNA replicates? You should know this for the exam. I'm going to ask about how does DNA replicate? And remember, the answer is B. because it replicates in a semi-conservative fashion, right? In a semi-conservative fashion. That means there's an old template strand left over and a newly synthesized red daughter strand. And do you remember, you should know for sure that prokaryotes divide by binary fission. Okay, bacteria, archaea, prokaryotes, right? They divide by binary fission. And remember the steps of binary fission. The circularized chromosome is copied, it moves to opposite ends of the cell, and then the cell divides into two. And do you remember, this is a type of asexual reproduction called binary fission. So clicker question number 3. Again, this question might appear on the exam because it's a clicker question. Why is binary fission an asexual process? Remember, it is because not A, right? Binary fission does occur in prokaryotes, but that's not why it's an asexual process. It does not require DNA replication. That's false. It produces two genetically identical cells, so it has to be C, right? Next, we talked about mitosis. Remember, mitosis is cell division in eukaryotes, which results in two genetically identical cells. You remember that? This is responsible for how eukaryotes, like multicellular eukaryotes, like you and I, it's how we grow, develop, and repair. However, if it's a unicellular eukaryote, then they can also divide asexually by mitosis. Do you remember our chromosomes are continuous pieces of chromatin? The chromosomes can be loosely associated, called euchromatin, or tightly packed up, called mitotic chromosomes. Do you remember that our DNA is wrapped around these proteins called histone proteins, forming nucleosomes? And that makes up our chromatin, Now, you should know a few things about the eukaryotic cell cycle. Okay, what do we need to know? First of all, you should know, do you remember which of these phases takes the longest? Interphase or mitosis? Which one takes the longest? Can you beat Wicket? That's right, Wicket. Interphase takes, on average, about 90% of the cell cycle, right? So you should know that interphase takes the longest. It takes 90% of the cell's cycle. And do you remember what happens kind of during each subphase? During G1, you should know that the cell grows. And what happens if the cell does not get permission to proceed into S phase? Do you remember what happens if the cell does not have permission to proceed into S phase? Do you remember what happens? Yes, the cell exits the cell cycle and enters the G0 phase. And then only when it gives the right checkpoint, clears, that's when, due to a signal, that's when the cell proceeds to S phase. And you should know what happens during S phase. Remember, the DNA is copied. The chromosomes are replicated. The DNA is copied during S phase. You can think of it as synthesis phase. Next, you should know what happens during G2. There's additional growth and preparation for division. Remember, that's when the centrioles get replicated. Next, we need to know what happens during each subphase of mitosis. Remember, prophase, prometaphase, metaphase, anaphase, telophase. What you should know is this. You should know that during prophase, the sister chromatid pairs condense and pair up. You see these X's are sister chromatids. Like, for instance, 2 copies of mom's chromosome 1, 2 copies of dad's chromosome 1, right? So you should know during prophase, sister chromatids pair up and condense. You should know that during prometaphase, the nuclear envelope breaks down and the sister chromatids begin to move to the center of the cell. Then you should know that during metaphase, the sister chromatid pairs line up at the center of the cell. Then you should know that during anaphase, you should know this. The sister chromatids separate from one another and start moving to opposite ends of the cell. And then lastly, you should know that during telophase, daughter nuclei form, right? Daughter nuclei form, and the DNA begins to decompress or uncondense. And you should know this. Animal cells undergo cleavage furrow formation for cytokinesis, while plant cells undergo cell plate formation for cytokinesis. And you should remember these facts. Mitosis results in 2 genetically identical cells in eukaryotes. And you should know this, that mitosis is the division of the DNA and the nuclei, while cytokinesis is the division of the actual cell. And here we are at clicker question number 4. In some slime molds and fungi, mitosis may occur without cytokinesis. So that means that the nuclei can divide without the cell dividing. What would you expect to see in these species? It would be B, right? Cells that contain multiple nuclei, because we're copying the nuclei, but we are not dividing the cell. So B would be the correct answer here. And then finally, we discussed a little bit about cancer cells, and I don't think I asked much about this on the exam. However, do you recall that there are checkpoint proteins, and these checkpoints are important. These various checkpoints are important for regulating how often a cell divides, and it's mutation of these checkpoint genes that results in cancer. And those include the proto-oncogenes and the tumor suppressor genes. All right, and that's about it, you guys. A pretty short exam. I think there's only 35 questions on this exam, so multiple choice and true false for my exam. I hope this was a good review for you. If you have any questions, drop them in the comment box below, and I wish you luck on the exam. I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.